Good morning. So we have an outstanding group of speakers. We've asked the speakers in the first session to help lay the groundwork by providing a foundation, by helping us refresh the state of the science. We have four speakers: Dr. Stephen Rosenberg, who's going to talk about personalized immunotherapy from the National Cancer Institute. We have Dr. Nair Rizvi from Columbia University, who's going to speak about immune modulators. We have Dr. Jay Berzofsky from the National Cancer Institute, who's going to help put in context work on therapeutic vaccines. And finally, Dr. Malcolm Brenner from the Baylor College of Medicine, who's going to speak about adoptive cell transfer. So each one is going to speak, and then all the panelists will be up here, and we'll save all questions for the end. So if the panelists want to come up, we'll get started.
I'll take that introduction. Well, good morning. I appreciate the fact that the organizers changed the order of speakers to enable me to go first this morning, as an indication of the interest that exists in immunotherapy now and the additional appropriations that appear to be coming to the National Cancer Institute. We have visiting in a few hours at the NCI 8 congressmen, including the head of the Appropriations Committee. And there were three areas that they asked to learn about when they come to the NIH, and one of the three was cancer immunotherapy. And so they'll be visiting my lab and learning some about this very exciting new field of immunotherapy today. I'm going to talk about T cells as a drug for the personalized immunotherapy of patients with cancer, and talk about cell transfer therapy, a kind of a treatment in which we can identify the specific cells that recognize cancer antigens for use in treatment. This kind of cell transfer therapy, or adoptive immunotherapy, has multiple advantages. One can grow easily on one shelf of a tabletop incubator now, 10 to the 11 cells that were highly selected to have a high avidity of recognition of tumor antigens. So numbers can be achieved in terms of the number of cells required for treatment. They can be administered activated so that they're entering the body with guns blazing. They're no longer tolerized or energized and can exhibit their anti-tumor effector functions immediately. Because you have the cells in a test tube, you can identify the exact cell subpopulations and other effector functions that are required to see cancer regression. And perhaps most importantly, this is the only approach to immunotherapy in which you can freely manipulate the host prior to cell transfer to provide an altered microenvironment for the cells, simply because the cells used for therapy have been removed from the body before agents are given to inhibit regulatory T cells, myeloid-derived suppressor cells, and other cells that can negatively impact on immunotherapy. Now, this cartoon shows the basic outline of the treatment that we use with some variations. We start by excising a tumor. We have a variety of techniques for growing multiple cultures from that tumor to grow lymphocytes, different lymphocyte cultures. Those lymphocyte cultures can then be in vitro assayed for specific tumor recognition to identify the exact lymphocytes that can recognize tumor. They are then expanded in vitro and reinfused into the patient. But very importantly, as shown by Mark Dudley back in 2002, a prior lymphodepletion of the patient prior to administering the cells can greatly enhance their anti-tumor activity, a lesson we learned in multiple animal models before attempting this in the human. Well, we've just completed a accrual with about a 3-year median follow-up of a trial looking at two different preparatory regimens prior to giving cells, either a non-myeloablative chemotherapy of cyclophosphamide and fludarabine that reduces circulating lymphocytes to zero for about 6 or 7 days before the immune system naturally reconstitutes, or that same preparative regimen with the addition of total body irradiation. There was no difference between these two, but notice especially the complete response rate with the non-myeloablative chemotherapy as a preparative regimen, the one that we have used routinely. The complete regression rate is 24%. And notice there are 5 additional patients with ongoing partial responses beyond 2 years. If you look at the conglomerate of these 24 complete responses in 101 patients, only one of these complete responses has ever recurred, and the others are ongoing anywhere from 14 to 53 months, which is a hallmark of immunotherapy. If you can induce a complete response either with IL-2 or cells, it's very likely to be durable. And in fact, as you can see now in this one particular trial, there appears to be a leveling out of the curve. This is out at 4 years in patients with metastatic melanoma, a little bit in excess of 50%. And so a 50% survival at 3 to 4 years in patients with the widely metastatic melanoma refractory to other treatments represents part of why we're so excited about immunotherapy, although this is in patients with metastatic melanoma, a tumor, of course, uniquely susceptible to this kind of approach. If you take the original 93 patients that we reported over the last 8 or 9 years and combine it with this 101 patients, that's 194 consecutive patients that we've treated with the same kind of approach, growing cells from fragments. And notice the overall complete regression rate is 23%, 44 patients, but especially notice that only 2 of these 44 patients that had a complete regression have ever recurred, 19 and 27 months. 42 of the 44 are complete responders, are ongoing anywhere from 14 out to over 10 years, and are very likely cured of their melanoma. And so adoptive cell therapy can mediate complete, durable, and likely curative regression in patients with widely metastatic melanoma. Now, what do TIL recognize that enables the in vivo destruction or control of the last cancer cell? And can this supply clues to the immunotherapy of the common epithelial cancers? Cancers of the colorectum, the esophagus, the pancreas, the ovary, the prostate, cancers that start in the epithelial lining of organs that today account for over 90% of all cancer deaths in this country, expected to reach about 600,000 deaths in this country alone this year. Well, we've begun attempts now with the revolution in genomic deep sequencing. One can perform whole exomic sequencing and look at the sequence of every gene, expressed gene in the body using whole exomic sequencing over the course of 2 days and obtain all of the mutations that exist in a cancer patient's malignancy. Now, one of the questions, of course, is since mutations are new or unique to cancer cells, they're different, they're non-self, can they be antigens? And very important to understand that although there may be multiple mutations, for a mutation to be a cancer antigen, it has to fulfill 2 very unique properties. First, it has to be processed intracellularly into a 9 to 11 amino acid peptide, and that peptide has to then be transported through the endoplasmic reticulum and fit on the surface of one of that particular patient's MHC molecules, either class 1 or class 2. One of the basic tenets of immune recognition by a T-cell receptor. A peptide has to fit in the groove of a class 1 or class 2 molecule on the surface and thereby be recognized. And so a mutation has to have the binding properties to fit in that particular unique patient's MHC molecules. And what might be an antigen in one patient might not be an antigen in another because the peptide doesn't fit in the groove of the particular MHC molecules that they have. So a very important biologic point to remember as we begin to search for personalized therapies for patients with cancer. Well, there are many prediction approaches to identify immunogenic epitopes, which are quite incomplete. There are many hundreds of class 1 and especially class 2 MHC molecules that can present these mutated peptides. Many of these are rare. Prediction algorithms are inaccurate, and especially inaccurate for antigens, peptides that are presented on class 2 molecules that are CD4 restricted. And so we developed and published in 2013 and have been continuing to improve and modify a different approach that we call a Tandem minigene approach, which is a method that can simultaneously evaluate the immunogenicity of all of the mutations in any individual cancer. That can range from a few dozen up to many hundreds. Can evaluate the immunogenicity of every mutated cancer peptide and their ability to be presented by all of the patient's MHC molecules simultaneously using autologous antigen presenting cells. So there are no predictions that are necessary. Everything is a direct measurement of the ability of an antigen to, ability of a mutation to be an active antigen. So this is the blueprint for how one might develop an immunotherapy for virtually any cancer. The blueprint involves resecting a tumor and isolating the genomic DNA and RNA. One can then do whole exomic sequencing, as well as transcriptome sequencing, RNA-seq, to identify all of the mutations that are in that cancer patient's genome, and by transcriptome analysis, all of the antigens that are actually expressed in that particular cell. And this can be done in the course of about 7 days, a couple of days to do the actual sequencing and 3 or 4 days to do the bioinformatic analyses. One can then, with this information, synthesize what we call a minigene, which is a mutation encoded in a particular 25-mer peptide. If one now makes a peptide and puts the mutated amino acid, the non-synonymous mutation, in the middle of a 25-mer, then that 25-mer, just illustrated here, flanked by 12 amino acids, contains every possible 9, 10, or 11 amino acid, including this mutation, right? The mutation can be the last amino acid, in which case this would be the peptide. It can be in the middle, or it can be the first. So this 25-mer contains the sequence of every possible mutation, every possible peptide containing this mutation. Well, what we do is we synthesize those 25-mers, and we string them together, the genes encoding them, into what we call a tandem minigene. So we have a sequence of 25-mer encoded peptides, one after the other, in a single DNA construct. We then in vitro transcribe it to RNA and insert it into the patient's own antigen-presenting cell, which is a lot smarter than any prediction algorithm, because the antigen-presenting cells, dendritic cells, contain all of the class 1 and class 2 molecules that can present a peptide. And so every possible peptide is inserted now into the patient's own antigen-presenting cell, and we can now use these tumor-infiltrating lymphocyte populations that we've grown to test whether any of the lymphocytes are recognizing any mutation present in this antigen-presenting cell. So again, we've encoded now this 25-mer with every mutation, and we can do this whether there are 20 mutations or whether there are 200 mutations. So here another example. This 25-mer peptide is in one of these mini-genes. They're strung together into what we call a tandem minigene. We then in vitro transcribe RNA, because it's very easy to electroporate and get expression at very high efficiency of RNA into antigen-presenting cells. And now we have a cell we can target to see if any mutations were immunogenic. So the advantages of this approach: no predictions necessary. Every candidate peptide and every MHC loci are included in the screen, and you don't need the tumor cell at all, because by stringing all the mutations together, you have basically a substitute for the entire cell if one's looking for immunogenic mutations. Well, this is one of the first patients that had a complete regression, one of those 42 patients with an ongoing complete regression out many years. This patient had 71 non-synonymous mutations, that is a mutation that changed an amino acid sequence. We constructed those into 12 tandem minigenes containing about 6 or 7 individual minigenes, and you can see one of those tandem minigenes here. We then just simply took the patient's lymphocytes that had mediated a complete regression of the patient's cancer, incubated with the antigen-presenting cells that have now had included every one of these tandem minigenes, and in this particular patient, only the first tandem minigene is recognized. The autologous tumor is recognized, of course, but only this first tandem minigene. Well, that encoded these 6 different minigenes, encoding 6 different mutations. To determine which mutation is actually the mutation, the specific mutation recognized by the T cell, what we do is back mutate each of the mutations one at a time back to wild type. Well, if all of those 6 were mutated, that antigen presenting cell is recognized. If we take this first antigen and mutate it back to wild type, still recognized. But if we mutate the second antigen back to wild type, all reactivity is lost, thereby identifying this particular protein, the kinesin family member 2C, involved in the stability of the mitotic signal, as the exact peptide and protein recognized by this patient's TIL that mediated a complete regression. Now, I'm going to show you a fair number of a couple of dozen slides today, but this is one of the most important of these slides and turned out to be quite a surprise when we conducted this kind of study. In 25 consecutive patients that had complete regressions of their cancer, the TIL that mediated the complete regression recognized 64 somatic mutations. Some TIL recognized 10 mutations, some just a single mutation. But every mutation recognized by a cell that mediated a complete regression was completely different, was unique to that particular patient, and in fact straddled the entire genome of that particular patient's expressed genes. And these are just random genes that happen to mutate and have the kind of properties that could present that mutation on an antigen-presenting cell and be recognized. And so we can take our preliminary conclusions a little farther and say that adoptive cell therapy can mediate complete, durable, and likely curative regressions of metastatic melanoma based on the recognition of immunogenic cancer mutations, mutations recognized by TIL that mediated a complete response. Well, I'd hypothesize that the recognition of these random somatic mutations is the final common pathway explaining cancer regression from probably all cancer immunotherapies that don't involve the genetic manipulation of the T cell. And so immunotherapies like IL-2, like anti-CTLA-4, there's already evidence that they're recognizing that they're unleashing recognition against the mutations, anti-PD-1, anti-CD4, and certainly tumor-infiltrating lymphocytes, all appear, as studies are now ongoing, and this whole idea is a couple of years old, all appear to be recognizing now mutations that occur in random proteins that exist in the cell, and every patient is recognizing something different, at least in that first 25. Well, can we utilize this approach now to treat the common epithelial malignancies that I mentioned kill over 90% of all people in this country, around the world, that die of cancer? Can we treat common epithelial cancers by targeting the unique immunogenic mutations expressed by those cancers? Because every cancer, of course, has multiple mutations. Well, melanoma has the largest number of mutations, all the way on the right here, but there are the gastrointestinal cancers that we'll discuss now that have fewer mutations, pancreas much less. This is a log scale, and this conglomerate of cancer mutations in patients with a variety of gastrointestinal cancers like those of the esophagus, the stomach, and the colorectum. Well, one of the first patients that we attempted to use this tandem minigene approach is this patient, Melinda, a 45-year-old female who had a metastatic cholangiocarcinoma. This is a carcinoma of the bile ducts in the liver. She had had a right hepatectomy. She had multiple lung and liver metastases. She got chemotherapy that can cause minor regressions for a few months, but is ineffective. No patients like this are cured. More chemotherapy, progressive disease. We resected a lung lesion for TIL. We infused them. It had minimal impact at all. A slight, did not make RECIST criteria, a slight prolongation of what we would expect for stable disease, but not terribly significant. We then utilized this tandem minigene approach to try to identify the exact mutated antigens that were recognized by T cells in this patient. Well, she had 26 different mutations, which enabled us to make 3 tandem minigenes, each encoding about 12 amino acids, 12 mutated proteins. And again, only tandem minigene 1 was recognized. This is OKT3, just stimulating all cells. In a co-culture assay, you can do it by ELISpot assay or just by interferon gamma release measured by ELISA. Well, tandem minigene 1 was recognized, and interestingly, the cells that recognized it were upregulating OX40 and much less Formin BB, suggesting that this was likely to be a CD4 cell that was recognizing this patient's mutation. Well, one by one, we reverted these mutations back to wild type, and only when we mutated this particular minigene back to wild type, the ERB2IP, was all reactivity against the T cells in the TIL lost, thereby identifying ERB2IP as the antigen recognized by this patient's T cells. Well, we then obtained cultures that were highly expressing T cell cultures that were highly recognizing this particular mutation. Over 90% of the cells in this culture could recognize this unique mutation in ERB2IP in the patient's tumor. And when we treated the patient with that unique population of highly effective cells, we see an ongoing regression of lung and liver metastases in this patient, now ongoing beyond 2 years. And you can see what happened here beginning in October 2013 when she was treated, and now 2.5 years later. You can see these large lung lesions, which were rapidly growing, have virtually disappeared. There's still a scar there. This lesion appears to be gone. This large lesion, this is still shrinking, is present in the lung, but could be scar, we don't know. This lesion, again, shrunk dramatically. These 2 large lesions have disappeared, and multiple liver lesions have disappeared. This is a hollow PET negative area in the liver. And so a substantial tumor regression for what previously was considered an untreatable common epithelial cancer. Well, here again, I think, is one of the most important lessons from this kind of work, in that we've now identified the exact mutation in 22 patients with a variety of common epithelial cancers. You can see cholangiocarcinomas, colon cancers, rectal, pancreatic, esophageal, lung, ovarian, cervical cancer. And in these 22 patients with common epithelial cancers, there were 57 somatic mutations that were recognized by the tumor-infiltrating lymphocytes present in this patient. And again, every one was different, a different intracellular protein, and none, in fact, overlapped with the melanoma mutations, because we have 23,000 at least expressed genes, and any one of them can become a cancer antigen is the lesson. Well, all were unique with the exception of one antigen. And that one antigen was KRAS, which is one of the most commonly expressed mutations in all of oncology. And you can see this patient, 3995, and this patient, 4095, recognized a KRAS mutation using this tandem minigene approach. And the first patient that we attempted to target with this KRAS mutation was a woman with a metastatic colon cancer expressing this mutation. She had had a sigmoid colectomy, invading the liver, a partial cystectomy, and had multiple lung metastatic deposits, which were growing. She had radiation therapy to the bladder suture line. Chemotherapy did not respond. We grew 2 lung metastases for TIL, purified a highly reactive population, and used this TMG approach to see if we could target the unique mutations in this patient's colorectal cancer. Again, this blueprint, this patient had 61 mutations. We made 5 TMGs, put it into the antigen-presenting cells, and checked with lymphocyte cultures from that patient. And we could use a variety of peptides or back mutate the individual genes. Now these 25-mers that we could pulse from that identified tandem minigene onto the patient's own antigen-presenting cells, and sure enough, could identify KRAS as the mutation recognized by that patient's tumor using either ELISpot techniques. So mutated KRAS is being recognized by that particular patient's culture. And we could identify the exact restriction element by taking COS cells, putting in each of the class 1 molecules, or 6 class 1 and 6 class 2. We knew this was expressing interferon gamma upregulating 401B, so we suspected that this one was class 1 restricted, recognized by CD8 cells, and I identified the C0802, a relatively rare class 1 antigen expressed by patients for which there are poor prediction algorithms, but we don't need them here, as the restriction element presenting this KRAS peptide in this patient. That's present in about 8% of Caucasians and African Americans. We could also identify the exact beta clonotype that these T cells were exhibited, that were recognizing the KRAS mutation, and could select a culture that had over 70% reactivity against this KRAS. It was present in only a tiny percent of the total TIL population. And when we treated this patient, the only patient we've treated with KRAS cells for which we have follow-up, you can see this dramatic reduction of the lung metastases in this patient, thereby showing that by attacking KRAS, we can mediate regression by targeting the unique mutation in those rare patients that express KRAS G12D and CW08. And as you can see, KRAS, even with this relatively rare beta clonotype, KRAS is so predominantly expressed in a variety of tumors that many thousands of patients that develop any one of these epithelial tumors might be treated with a KRAS T cell receptor. We've identified that T cell receptor for insertion into a patient's own normal lymphocytes. So I would just emphasize before we move on that this is a blueprint that one could potentially apply to any cancer type, because we can perform exome and transcriptome sequencing. We can grow TIL cultures, and we're looking at improved ways to grow them, introduce them into a patient's antigen presenting cell, and attempt to identify the mutation recognized. Well, the reagents needed for a treatment like this are, of course, the ability to sequence mutations, and you can sequence them right off the primary tumor, right out of paraffin-fixed sections to look at whole exomic sequencing. More difficult to do the transcriptome sequencing from these fixed cells. And you need T lymphocytes that can be grown. Everything we've done up until now has been from fresh tumor, but that's often not available or difficult to obtain, since most patients with metastatic cancer have these cancers in visceral organs. And so we've looked to see if we could find ways to enrich for these tumor-reactive cells that are recognizing mutations. And Elena Gross has shown that the PD-1 molecule, the molecule that is being attacked by checkpoint modulators and is also a molecule that is upregulated as a T cell continues to get stimulated, it's considered an exhaustion molecule. You can see tumors express about 25% of their lymphocytes as a median will express PD-1. In peripheral blood, much less, only about 2 or 3%. Well, Elena, a postdoc fellow in the lab, showed that all of the anti-tumor reactivity in tumor-infiltrating lymphocytes is in the PD-1 positive population. She looked at other markers as well, but we'll concentrate on PD-1, and virtually no reactivity in the PD-1 negative cells, which makes some intuitive sense, because if a lymphocyte inside the tumor is being repetitively stimulated by a tumor antigen, it's going to upregulate anti-PD-1 as an exhaustion marker. And that's true for every tumor that we've looked at. These are all melanomas. Well, we wanted to see if we could identify cells in the circulation so that we wouldn't have to resect tumors to get these lymphocytes. And only about 2 to 3% of all of our circulating lymphocytes in peripheral blood, we have 1 million lymphocytes for every milliliter of blood, only about 2.5% express anti-PD-1. And sure enough, it turns out that in the peripheral blood as well, all of the anti-tumor cells in peripheral blood are in that 2 to 3% of cells that express PD-1, and not in the PD-1 negative cells for each one of these patients. And so we can find these anti-tumor cells in peripheral blood, and now use the Tandem minigene approach to see which of these cells can recognize tumor. In this case, a patient that had melanoma, that had Tandem minigene 9 recognized by lymphocytes. Among these 28 Tandem minigenes in the multiple mutations present in this patient, we could, by looking at 4-1BB, look at upregulation of 4-1BB to identify a high population, 89%, that recognize that Tandem minigene. And in fact, when we now explore the patient's peripheral blood lymphocytes for recognition of Tandem minigenes, there are 3 that are identified, and each 3 are unique antigens involved in microtubule movement, in intracellular trafficking of molecules, and RNA splicing. Again, everyone an individual unique protein identified by lymphocytes that are circulating in peripheral blood. And so our preliminary conclusion is that the selection of CD8 PD-1 positive lymphocytes, not only from TIL, but from peripheral blood as well, enables the direct enrichment of tumor-reactive cells. The peripheral blood cells enriched for PD-1 can be screened using this Tandem minigene approach. And then when mutation-reactive T cells are recognized in TIL, these are most often in the PD-1 positive population, and again, in every patient, unique. We've now tested, for example, 26 patients with metastatic colorectal cancers. In 23 of those 26, we could find immunogenic mutations recognized by T cells in that patient. Well, I leave you with the following tentative conclusions, and that is that cell transfer therapy can mediate durable regressions in patients with metastatic cancer refractory to other treatments, that T cells that recognize unique somatic mutations can be found in TIL and PBL, and that identification and targeting of mutations unique to each cancer has the potential to extend cell therapy to patients with common epithelial cancers. But the problems are daunting, because we have to basically develop a new drug for every patient. And that drug has to recognize a mutation unique to that patient, binding to that patient's unique surface MHC molecule. So there are multiple steps involved in this, but we can go from resection of a tumor, although now we can go from peripheral blood to cells available for treatment in about 5 to 6 weeks for an individual patient. And I think this highly personalized approach is now being studied and somewhat endorsed by a variety of biotechnology companies that see this as a possible means for application to a wide variety of cancer types. Well, thank you for your very kind attention.
Thank you to Dr. Rosenberg. We'll Dr. Rosenberg has another very important engagement, so we'll take a few questions for him.
That was great talk. I might have missed this. Did you actually compare the peripheral PD-1 positive cells and the central one and see whether they both actually recognize?
We have several patients in which we've looked within the TIL, within the tumor for reactivity, as well as in peripheral blood. And almost always when you find them in TIL, but not always, you find them in peripheral blood. And we have at least one example where we found them in peripheral blood and not in the tumor-infiltrating lymphocytes. So the two overlap to a significant extent.
Antigens identified CD4 reactive or CD8? And the second question is, were both of your KRAS CD4 reactive that you found? And finally, should we be banking viable cells from all of our patients that undergo surgery now in anticipation of possibly using this approach?
KRAS in the two patients were both class 1 restricted, recognized by CD8 cells. But if you look at the panoply of different cells and mutations that are reactive, they're both CD4 and CD8, perhaps a slight predominance of CD8, but many CD4 class 2 restricted T cells identified as well. I think it would be quite reasonable to obtain fresh tumor and store it in a cryopreserved fashion, keeping those tumor cells alive from every patient who has a resected cancer, because eventually those TIL might be identified for use in cell therapy. But certainly peripheral blood should be stored for a variety of reasons, not least of which that they also contain, that peripheral blood also contains T cells recognizing mutations. So I would say the storage of these biorepositories will be very important. But of course, we'll always have the potential opportunity to take a patient sitting across from us and obtain the reagents we need from that patient to develop this kind of personalized therapy. Dr. Millet.
Someone can take TIL and test them against the HLA match. So I see peptides. In other words, you may not find that TIL recognize individual peptides, right? So your thoughts there?
So why can you see shared reactivity when everything is unique? Well, there are also lymphocytes, especially in melanoma, but not in many, there are equivalents in other tumors, but not often, that recognize shared melanoma melanocyte antigens like MART1 and GP100 that were identified over 20 years ago. However, when you take very highly avid T cell receptors against those melanocyte antigens, their anti-tumor activity is very weak, and it mediates destruction of melanocytes in the eye and in the ear and in the skin. There's no off-tumor, on-target toxicity when you use these TIL that mediate cancer regressions, because of course you're targeting something that's a mutation only in that cancer and not in normal cells. But there are shared reactivities for many years. We've tried to target those shared reactivities unsuccessfully, like cancer testis antigens as well, without much success. And it's probably the reason why cancer vaccines that have tried to target these shared, relatively ineffective antigens have not been effective at all in cancer treatment, because they're not targeting the unique mutation in that patient's tumor. Dr. Chapman.
In a sequence of patients, how many, all of this relatively complex analysis, just from a practical standpoint, at this point in our knowledge, how many patients will end up with a therapy that will be specific for their tumor?
So we're working hard, not only to identify all the antigens, which we can do now, but to identify even the rarest of populations that can recognize these antigens. And we now have improved methods to grow them by doing a clonal analysis right out of the tumor and using high throughput immunologic screening. So it's a very hard question to answer. What I can tell you is that in an analysis of a whole variety of gastrointestinal cancers by Eric Tran, a fellow in the lab, he's now analyzed 26 of these, and in 23 could identify T cells that recognize mutations in that patient's cancer. Now, how many will be therapeutic? We don't know yet. Ideally, one would want to recognize more than one antigen at a given time, and there are many ways that we could improve the growth of these cells to keep them from getting differentiated by inserting AKT inhibitors or by growing them in IL-15 and IL-21. So there are a lot of potential improvements, but basically you can run through this blueprint in about 5 to 6 weeks for any cancer patient.
So is there any one step that's blocking the clinical trial at this point?
Well, there are one biologic and one technical. The biology, and we're at the mercy of the biology. If one of those mutations will bind to one of the patient's MHC molecules, there are 12, 6 class 1 and 6 class 2, which is likely, then that patient will be susceptible, potentially susceptible to the therapy. The technical block is identifying the rare lymphocytes. We have one lymphocyte that recognizes these mutations that is 0.02% of the circulating T cells. So 2 10,000ths of 2 out of 10,000 cells, but we're looking at ways to identify those very rare cells using high throughput sequencing. I think it should be very possible. There's a complex technique, but it's pretty straightforward once one mechanizes it. And although it's complex, if we have to find treatments that'll induce durable complete regressions, it's a little too high a bar to ask for to have it be simple.
Sorry, I have to keep us on time. I've got one very patient waiter in the back row, and then Dr. Brzezowski, 2 more questions, and then we'll have to move on. Sorry.
Thank you very much. Wonderful data. Thanks for sharing this. I was wondering, for the Tandem minigene approach, I guess a prerequisite would be that the antigen processing machinery is the same in the APCs as the tumor cells. I was wondering, when you look at epithelial cancers and melanoma, how closely they actually match with the tumor cells, because there could be different ratios of immunoproteasome to proteasome, et cetera.
The immunoproteasome can certainly process antigens that are not presented necessarily in the cancer, but the overwhelming majority are presented in the cancer. And because we not only look at the exomic mutations, but we do the transcriptome analysis, the RNA-seq, we can selectively look at just those proteins that are actually being presented in the cell. That is DNA to RNA to protein expressing that mutation. So we can narrow down the possibilities by doing RNA-seq. But virtually in virtually every example that we've looked, the antigens recognized on the antigen presenting cell are also seen on the tumor when you have the tumor cell line available, which is not frequent. Yes.
Steve, beautiful talk. Now that you can map these somatic mutations that are recognized by TIL, have you had a chance to look at TILs from patients who've had long-term, durable, complete responses to find out whether those have a higher probability of having TILs specific for more than one mutation? Because the prediction would be that if there's only one mutation that's targeted, escape may be higher probability than if the TIL are targeting multiple mutations. And if that's the case, then you might need to combine T cells for adoptive therapy against different mutations. But you can test that with your TIL, because presumably they give more durable responses.
I think you raise a very good point. There appears to be a correlation between the number of mutations that are present on exome analysis and the number that we can recognize. And as you know, with respect to the checkpoint modulators, anti-PD-1, there is a relationship between the number of mutations and the likelihood of having a response in patients with smoking-induced lung cancer. So that correlation does appear to exist. Patients that have mismatch repair mutations have a lot of mutations. They're also susceptible to anti-PD-1. So this all fits with the hypothesis that the final common pathway of cancer immunotherapy is the recognition of cancer mutations. Thank you.
Rosenberg, thank you.